Protein synthesis by Helen, Mahsan, Saba, and Emma. The first step in protein synthesis is transcription. This is where the enzyme called RNA polymerase creates a complementary copy of the code by building a molecule of mRNA. In this picture, we have a DNA double helix being unwound or unzipped by the enzyme called helicase. As you can see, we have color-coded the bases adenine, thymine, cytosine, and guanine. There is the sense strand or coding strand that is the segment of double-stranded DNA running from 5' to 3' that is complementary to the antisense strand. The sense strand is the strand of DNA that has the same sequence as the mRNA, which takes the antisense strand as its template during transcription and eventually undergoes translation into a protein. The DNA double helix unwinds and the hydrogen bonds are broken at the site of the gene being transcribed. At this site, there is a pool of free nucleotides. Then, one strand of DNA, the coding strand, is used as the template for transcription by complementary base pairing. In RNA synthesis, it is uracil which pairs with adenine, instead of thymine. An exon is any nucleotide sequence encoded by a gene that remains present within the final mature RNA product of that gene once the introns have been removed by RNA splicing. Once the mRNA strand is formed, it leaves the nucleus through pores in the nuclear membrane and passes to ribosomes in the cytoplasm, where the information can be read. Now we are moving on to the second step in protein synthesis, which is called amino acid activation. Also, a note about the genetic code. It is a sequence of bases in nucleic acid, and it dictates the order in which specific amino acids are to be assembled and combined to make proteins. The code is a 3-letter or triplet code, meaning that the sequence of 3 bases stands for one of the 20 amino acids and is called a codon. Some of these codons represent the punctuations of the code, meaning that there are start and stop triplets. In this stage, amino acids are activated by combining with short lengths of a different sort of RNA, called tRNA. tRNA translates a 3-base sequence into an amino acid sequence. This activation occurs in the cytoplasm. All of the tRNAs have a cloverleaf shape, and there is a different tRNA for each of the 20 amino acids involved in protein synthesis. At one end of each tRNA molecule is a site where one particular amino acid of the 20 involved can be joined. At the other end, there is a sequence of 3 bases called an anticodon. This anticodon is complementary to the codon of mRNA that codes for the specific amino acid. The amino acid becomes attached to its tRNA by a specific enzyme in a reaction that also requires ATP. These enzymes are specific to the particular amino acids and types of tRNA to be used in protein synthesis. The specificity of the enzymes is a way of ensuring the correct amino acids are used in the right sequence. The third and final step of protein synthesis is translation. In this final stage, a protein chain is assembled one amino acid residue at a time in tiny organelles called ribosomes. These move along the messenger RNA, reading the codons from the start codon. In the ribosome, for each mRNA codon, the complementary anticodon of the tRNA amino acid complex slots into place and is temporarily held in position by hydrogen bonds. While held there, the amino acids of neighbor tRNA amino acid complexes are joined by a peptide linkage. This frees the first tRNA, which moves back into the cytoplasm for reuse. Once this is done, the ribosome moves on to the next mRNA codon. This process continues until a ribosome meets a stop codon. The end.